I'm Stina Troyer here to present on behalf of a team who did an experimental oyster restoration project in the Salish Sea. For a little bit of background, you should know that restoration of Olympia oysters is a priority in Washington State and the greater Salish Sea, and that previous studies have confirmed shellfish in the Puget Sound have accumulated a freshwater toxin or microcystins that have breached the land-sea interface. So the goal for this project was to identify the viability of an Olympia oyster restoration at a South Sound site, as well as to assess the human health risks associated with climate change impacts, as those harmful algal bloom events are expected to expand kind of in both scale and in intensity with global climate change. And then, of course, our goal number 3, teamwork makes the dream work, so we're going to do a lot of collaboration. Starting with that third goal, collaboration, I do want to begin with noting our incredible team of volunteers who made this project happen. It really does take a village to do all the work that was included with this project, and I have to shout out my mom and my collaborator's daughter. I really think we could do a whole presentation on the people who helped make this work happen during a global pandemic of all things. Another important part of the team is, of course, the funders. So this is a project that was funded with money from the EPA through the National Estuary Program, as well as the Coastal Conservation Association, Rose Foundation, and Pierce County. I will say that the National Estuary Program part of the grant was unique in that it was administered through the Washington State Department of Health to the Pierce County Surface Water Management Team, who then divided the grant up between Harbor Wildwatch, which is the small education nonprofit that I work for, and the Tacoma-Pierce County Health Department. Together, we split that project in half, where the Health Department monitored for harmful algal blooms, and Harbor Wildwatch collaborated with Mike Behrens, who's a Pacific Lutheran University professor, to do more of the oyster restoration side of things. I will say my favorite part is that we got to pay students from both PLU and UWT to help with this project, and all of this research took place at a Key Pen Park and a Washington State Park. So where are these sites, you might be wondering? Well, come along with me on a journey to the Pacific Northwest. You can see here in the middle there's a picture of the Salish Sea, which is a place that encompasses waterways from Canada as well as Washington. And you'll see down in the lower part is the Puget Sound, which is the southern body of water there, south of Seattle, across from Tacoma, out on the Key Peninsula, nestled into Carr Inlet, and that's where you'll find our study locations, which include Penrose Point State Park, Maple Hollow, and Bay Lake. Conveniently, all of these places are sites that our organizations are familiar working with. The health department has done a lot of monitoring over at Bay Lake and has previous documents or studies about harmful algal blooms that have occurred in that system. You see on the map that Bay Lake connects nicely over to Mayo Cove, which is where Penrose Point State Park is located. Penrose is a place with easy public access. It's also a really popular site for harvesting shellfish, which obviously makes it an ideal spot for this study of the interaction between freshwater hazardous algal blooms and marine shellfish. And then, of course, Penrose and Maple Hollow are both sites that Harbor Wildwatch has done a lot of long-term beach monitoring. So we have the data to do a before-after control impact analysis. Now that you know where we are, we can go ahead and take a look at that first project goal, which is how do freshwater harmful algal blooms impact nearby shellfish populations? Here's the deal. You don't get to know. While previous studies in Mayo Cove confirmed the accumulation of those freshwater microcystins in the Puget Sound shellfish using the mussel cages that are nicely pictured here, You can imagine that when you don't have either enough water flow during the study time frame connecting a harmful bloom at Bay Lake to Mayo Cove, or maybe the levels of microcystins were too low for detection, you don't get any data. While it feels odd to have hoped for a harmful algal bloom, I think it is a good thing that shellfish and human health were not impacted during this time, but because there was no hazardous algal bloom interaction, the rest of this presentation will focus on the oyster restoration side of the story. Yes, the important Olympia oyster. This is Washington's only native oyster species. They filter up to 2 gallons of water per day. So I would say oysters are pretty vital for healthy, clean water. They're also great habitat builders. So the oyster beds, all those shells create reefs, which provide habitat for all sorts of marine invertebrates to thrive in. Those marine invertebrates are in turn food for things like the iconic Pacific Northwest salmon, and really just I'm thinking of oysters as this foundational player in a healthy ecosystem. And here's the thing. They're delicious, which is why, unfortunately, they've declined here in the Pacific Northwest. During the California Gold Rush, if you struck it rich, the thing to do was order the most expensive meal on the menu. And in many cases, this was the bacony, oystery, omelette-y thing called a Hangtown fry. And while, like San Francisco, at some point had its own populations of Olympia oysters, they ran out, and so they started shipping them from places like the Pacific Northwest, which, with the advent of steamboat and rail, we could ship live oysters down to California where they could be consumed, and thus we see the overharvesting of Olympia oysters in Washington state. Looking here at the map on the left, you can see that historically there were many Olympia oyster beds in the Puget Sound, and now, looking to the right, there are many great groups doing restoration work to try and restore those missing oyster beds. This leads us to the second goal of our study, which is determining if we can successfully reestablish Olympia oysters in Carr Inlet. This question comes in 2 parts, the first being, are there sufficient larvae to naturally recover the site? The photo here shows the if you build it, they will come approach, with the idea that if the ideal habitat exists at a beach, then the naturally occurring juvenile oysters will settle out and recruit onto your location. And that's, of course, assuming that there's a viable reproducing population of Olympia oysters. To look for natural recruitment, we monitored both Penrose and Maple Hollow in the summers of 2019, 2020, and 2021. Each of these sites had 7 stations, each with 3 replicate shell strings installed at the 0-foot tidal height. And then every 2 weeks, May through August, during those summers, these shells were swapped out, and the 210 shells per site were observed for the presence of juvenile oysters using dissection microscopes. And here's the spoiler alert. We counted so many zeros. You can see circled in pencil here one of the few juvenile oysters that we observed during the many summers we did this, but overall the oyster settlement was extremely low at both sites with only 0 to 3 recruits per summer. To answer our first question about oyster restoration, no, there is not sufficient larvae to naturally recover Penrose Point State Park. So on to our second question regarding a more comprehensive restoration attempt of establishing shell habitat as well as measuring the survival and growth of outplanted juvenile oysters. To do this part, we established 5 test plots in September of 2020. Each one of those plots had about 1 cubic yard of Pacific oyster shell hash, which we spread in a 10 by 10 foot area. I have to say that was a lot of work and absolutely a pandemic highlight. But anyhow, each one of those plots got sprinkled with about 1,200 juvenile oysters, only about the size of a quarter. And then with those plots in place, we buckled into sampling both the movement of that additional shell material as well as the survival and growth of the juvenile oysters. First, we'll talk about what we saw with the shell movement. You can see from the photo of us in that original plot, exhausted it looks like from laying out all that shell. This nice neat 10 by 10 square definitely shifted over time. So then we're thinking, is it transport? Is the shell sinking in the mucky mud? What's going on here? And it turns out that the answer is both. The shell is sinking and being transported off of those squares. One of the ways we measured this was using a straight line measurement from the different edges of those plots. And you can see that the left and right graphs on the top indicate spread along the shoreline oriented with the observer facing the water, and then those high and low graphs on the bottom indicate the spread of shell across the tidal height, with the high part being looking up at the beach and then the low part looking towards the water. So that transport of shell material was definitely greatest at test plots 3 through 5, and we suspect that that had a lot to do with how the shoreline's oriented, as well as the lack of rocky habitat and vertical relief in that area of the beach. As far as looking at kind of the percent cover of shell material, we could see that that declined over all of the plots over time, with that average cover stabilizing around 40 to 70%. The test plot that we noticed with the most vertical relief in the rockiest area was the one that maintained most of its shell cover, which is important for our recommendations later. Now we want to look at the growth and survival of the outplanted juvenile oysters. So to assess how they did, we basically just counted and measured the oysters that we could find in 10 randomly placed quarter-meter quadrats on each plot. Now the results for this part were very dramatic. Even just 1 month into surveying, you could see that there's a severe decline in the density of the oysters. And that variation in oyster density that we see is likely due to the random sampling of those quadrats. You might notice that test plots 3, 4, and 5 have zeros, and then later increase, which we know is not due to recruitment. We also see that the oysters decrease in size and then increase. We suspect that there's some size-selective mortality going on at the beginning, perhaps the big oysters getting eaten, and then the surviving oysters starting to grow over time. So our next question was, are the juvenile oysters indeed being predated upon? We were able to add a predation study to the party, and we installed 5 predation stations adjacent to those oyster restoration test plots. Each of these stations had 4 different treatments with the 30 juvenile oysters. There was a fully enclosed cage treatment, a cage control that was partially open, and then 2 completely open treatments. One of those had the juvenile oysters sprinkled on top of the shell hash, kind of mimicking our previous methodology, and then the second open treatment was mixing those baby oysters into the shell hash to see if maybe that would help decrease the predation and increase the survivorship of those little guys. What we found is it turns out it doesn't matter how you mix and match your oysters. If they are not fully enclosed, they're going to be somebody's breakfast. So we initially suspected red rock crabs as the culprit, being that they seem to populate all this new shell material, but we've since added raccoons as another suspect here because we observed them foraging along the beach many of the nights that we were out surveying. So with all of this information, it turns out that shell stability and oyster survival and growth vary a lot. And this leads us to our recommendations, which would be that any future Olympia oyster restoration projects should definitely focus their efforts on adding shell habitat to areas of the beach with the greatest shell stability so it doesn't move around on you. And then because there are not enough oysters naturally recruiting to this area, we think that oyster seed should be added in a way that's mindful of reducing predation. Additionally, while we didn't get to see the impacts of harmful algae blooms on shellfish in Mayo Cove, This is still a great wonder, and I know that the health department continues to track and monitor the microsystems in marine shellfish because they want to have a better idea about the conditions that necessitate whether they need to post advisories to warn the public about the risks of eating shellfish in the event of a harmful algal bloom. And that's my time. Learn, have fun.